process in sexual reproduction that produces gametes with unique genetic material. Unlike mitosis, it goes through 2 rounds of division, producing 4 haploid cells, which are then used for fertilization. Meiosis starts with interphase. Interphase has 3 parts: G1, S phase, and G2. In G1, the cell produces organelles for DNA replication. During S phase, DNA synthesis occurs, replicating chromosomes. During G2, the cell prepares for meiosis by making proteins. During prophase 1, the chromatin condenses to visible chromosomes. Homologous chromosomes form tetrads, which cross over to exchange genetic material. This increases genetic diversity. The nuclear envelope disintegrates and the spindle fibers form. During metaphase 1, the homologous chromosomes line up in a random fashion at the center of the cell. Spindle fibers attach to the kinetochores.
The orientation of homologous pairs at the metaphase plate is random, which means that each pair can line up in either orientation. This process is called independent assortment, and it creates genetic diversity by producing different combinations of alleles in the daughter cells. The cell undergoes a checkpoint during metaphase 1 to ensure that all chromosomes are properly aligned at the metaphase plate before proceeding to anaphase 1. This checkpoint helps to prevent errors in chromosome segregation, such as non-disjunction.
During anaphase 1, the homologous chromosomes separate and move towards opposite poles of the cell. The sister chromatids remain attached.
Anaphase 1 is a crucial stage in meiosis, as it ensures that each of the daughter cells formed during meiosis 1 will have exactly 1 copy of each homologous chromosome. Errors during anaphase 1 can result in the formation of daughter cells with too many or too few chromosomes, leading to genetic abnormalities such as Down syndrome.
In telophase 1, the chromosomes arrive at the opposite poles and a new nuclear envelope forms around these chromosomes. The cell is then divided into 2 haploid daughter cells through cytokinesis.
In meiosis, cytokinesis usually follows telophase 1 immediately, resulting in 2 haploid daughter cells with half the number of chromosomes as the original cell. Cytokinesis is the process of the division of the cytoplasm to form 2 separate daughter cells. Telophase 1 is followed by interkinesis, a brief period of rest where the cell prepares for meiosis 2.
Prophase 2, the nuclear envelope breaks down and the spindle fibers form.
During prophase 2, homologous chromosomes do not pair up like they do in prophase 1, but rather the chromosomes line up randomly at the equator of the cell, which is known as independent assortment. There is no crossing over during prophase 2 as it occurred during prophase 1.
Chromosomes line up in the center of the cell. The spindle fibers attach to the kinetochores.
Metaphase 2 is also responsible for generating genetic diversity in offsprings, as each gamete produced by an individual will be unique due to the random orientation of chromosomes during metaphase 2. The cells produced after metaphase 2 are haploid, which means they contain half the number of chromosomes as the original cell.
During anaphase 2, the sister chromatids separate and move towards opposite poles of the cell.
The spindle fibers shorten as the chromatids are pulled towards the poles, causing the cell to elongate. Anaphase 2 is completed when the chromosomes reach the opposite poles of the cell and the spindle fibers are fully extended.
In telophase 2, the chromosomes arrive at the opposite poles of the cell, and a new nuclear envelope forms around each set of chromosomes. The cell then divides into 4 daughter cells through cytokinesis. Each daughter cell has a haploid set of chromosomes. As shown, the process begins with 1 diploid cell, which divides into 2 haploid cells in the first round of meiosis, and then into 4 haploid cells through the second round of meiosis.
Just to jog your memory, here's a quick recap of the whole process of meiosis. We hope this lesson was helpful for you.